Last part of our lecture on atoms and molecules is talking about acids, bases, and the pH scale. Okay, so let's talk first about what exactly pH is. From a mathematical standpoint, pH is equal to the negative log of the hydrogen ion concentration. And let's talk about that for a second. You should be familiar with what log is from your previous math classes, so I'm not going to worry about that. The hydrogen ion concentration is the number of hydrogen ions present in a given unit of, for example, water. And you'll notice that around the hydrogen ion symbol, there are these square brackets. Those square brackets mean concentration. So anytime you see a atom or molecule with those square brackets around it, that means the concentration of that atom or molecule. So here's the deal with the pH scale. The pH scale is kind of bizarre because it goes from a value of 0 all the way up to a value of 14. And that might seem a little strange, but if you understand how the pH scale came about in the first place, then you will understand why it goes from a value of 0 to 14. If we look at water, if I were to take a beaker full of water, okay, so here's my beaker full of water, and if I were able to look at individual water molecules in that beaker of water and take a picture, I would see that, well, first of all, before I even took a picture, I would notice that all the water molecules are moving around constantly. They're constantly vibrating and moving around and bumping into each other and clashing and banging and into each other and colliding. If I took a snapshot, a picture of what those molecules were doing at any given point in time, I would see that a small percentage of those water molecules would have split apart into a hydrogen ion and what's called a hydroxy ion. Okay, a very small percentage. In pure water, it turns out that the amount, the concentration of these hydrogen ions and the concentration of these hydroxy ions in pure water is about 1 times 10 to the negative 7th. That's a 0.1234567. So it's a very tiny amount of all the water in that beaker that is in the state of being split apart into a hydrogen and a hydroxy ion. Now, if I take this concentration, which is a concentration of hydrogen ions, it's also a concentration of hydroxy ions, but let's just focus on the fact that this is the concentration of hydrogen ions. If I take as the equation up here says, the negative log of 1 times 10 to the negative 7th, the number I get is 7. So 7 is neutral. This is on the pH scale, okay? Anything below 7 down to 0 is acidic. Anything above 7 up to 14 is basic, okay? What this means is anywhere from 0 to 7 is acidic. That means you have a lot of hydrogen ions present. Anywhere from 7 to 14 is fewer hydrogen ions, okay? So acid means it has hydrogen ions present, okay? Hydrogen ions present. Sorry, I've made kind of a mess on this screen, haven't I? Okay. So that's how the pH scale was created. Let's take a look a little more closely at it and look at acidic solutions. Remember, these are the ones that have a value of 0 up to just below 7. And again, remember, this is a measurement of how many hydrogen ions are present. We can look at things like pure water is neutral, that's at 7. Things like milk are only slightly acidic. Soda beverages, beer and vinegar are much more acidic at a 3. The acid in your stomach is at a 1. And by the way, that means that you have 1 times 10 to the negative 1 hydrogen ions, which means you have that many hydrogen ions present. So it's 10% hydrogen ions. That's a big concentration. Hydrochloric acid is 100% hydrogen ions. Much, much larger value. All acids at their most basic are solutions that can donate a proton. Remember that hydrogen is the simplest of all of the atoms. It contains a single proton, which is orbited by a single electron. So a hydrogen ion, if I take away its one electron here, and I'm going to erase it out for you. If I take away its one electron, all that's left then is the hydrogen ion. And as a result, it is literally a proton donor. It's able to give up its one protein to other things. Okay, now what about basic solutions? Basic solutions, by definition, have a very low amount of hydrogen ions, okay? So for example, they usually have, since they have a very low hydrogen ion concentration, they tend to have a relatively high hydroxy concentration. Lots of OHs. For example, human blood is slightly basic. It has a pH of about 7.3 to 7.4 roughly. Ammonia that you might use around your house to clean with has a pH of 11. And then sodium hydroxide is something that is extremely basic, okay? Has almost all hydroxy ions and very little to no hydrogen ions. Basic solutions are defined as solutions that can either take up protons or they can release those hydroxy ions, okay? Either way. They can take up protons, which is just another way of saying taking up hydrogen ions, or they can release hydroxy ions. Now, one of the things that we're going to talk about when you get into Bio 201, and particularly when we get into Bio 202, is this idea of buffers. Buffers are really important because these are solutions that have the ability to either take up protons when needed. So if a solution gets too acidic, a buffer can absorb some of the extra hydrogen ions so that it gets it back to less acidic, or it can release, or it can take up, I guess I should say, we'll do it this way, take up hydroxy ions, so that if it gets too basic, we can soak up some of those hydroxyl ions so that if the solution goes back closer to neutral. Okay, so they buffer things so that things don't get too acidic or too basic. These turn out to be extremely important in our blood. Our blood has to stay in a very tight pH range, and anything that causes it to sway too far acidic or basic can lead to death. For example, if your blood were to become completely neutral, exactly neutral at 7.0, that would be enough of a change in the pH of your blood to kill you. So we have lots of different buffers in the blood. We'll talk more about these actually when we get to Bio 202. But for now, just trust me that there are different buffers in the blood in the form of proteins and some other types of molecules that do indeed help to make sure that our blood pH stays within a very tight range. Congratulations, you made it through your first round of chemistry lectures. We've got one more big round of chemistry lectures coming next week, and then we'll be moving on some more into biology. See you then.